but we can't hear you.
How about now?
Now we can.
Great. Sorry, I have a new mic that I need to press a button on to make it actually work. So I'm Isaac. I'm a third-year grad student in the Starks lab, and I'm going to be talking to you guys about a project that I've been working on for really 3 years now, and that's self-organization and resilience in the honeybee extended phenotype. Oops. Like this. So the extended phenotype is a term coined by Richard Dawkins in a book with the same title. And essentially, it's the effect, he describes it as the effect that genes have on the environment. And I like to think about it as the effect that an animal's genotype has on behaviors that allow it to make constructions. So this is an example of a termite mound. So termites build these out of mud, and they then live inside them. Weaver birds build these really neat hanging nests out of leaf material. Humans have arguably the most complex extended phenotype where we build these really large cityscapes. This is the skyline of Chicago, which is my hometown. And I'm biased, but I think it's one of the most beautiful cities in the world. And of course, honeybees, which is the species that I'm interested in. When we talk about their extended phenotype, we're talking about the comb. So the comb is all of this stuff that you can see the bees standing on. And what's really interesting about honeybee comb structure is that it's arranged in a characteristic and repeatable way. So generally, honey and nectar, represented here in blue, and which are the colony's carbohydrate stores, are stored near the top and the edges of a colony. The brood, which is the subadult bees, so the eggs, larvae, and pupa, are stored in sort of a central aggregate in the middle and kind of towards the bottom of the colony. And pollen, which is the colony's protein source, is stored in a thin strip between the two. And honeybees are able to maintain this characteristic organization through self-organization behavior. So self-organization is a phenomenon by which animals use simple rules to lead to complex group-level patterns. So one example of this is flocks of sheep, or as Ben sort of touched on last week, schools of fish. So in this case, the sort of cohesion and directionality of this sheep flock is maintained, even though the animals aren't all communicating with each other. Even though a sheep up here is not directly interacting with a sheep back here, because they're interacting with the sheep around them, like I said, the cohesion and directionality of this movement is maintained. Another example is in living architecture, like this army ant bridge. So this is an experiment that was done by Helen McCreary, who's sort of a Starks Lab honorary member. And army ants build these bridges when there's sort of uneven terrain over where their foraging paths lie. So these bridges are maintained based on traffic. So when there's more traffic going over the bridge, more ants are likely to stop and remain in the bridge, causing it to grow larger. And when traffic over the bridge slows down, ants that are in the bridge are more likely to leave, causing the bridge to eventually dissipate. So it's able to sort of meet the demand of the flow of traffic. So how does this apply to honeybee comb arrangement? There's three sort of main rules that keep honeybee comb arranged the way that it is. First, the queen oviposits near existing brood. So the queen tends to lay eggs near where brood are already existing. And this helps maintain sort of a central cluster, central aggregate of brood. Second, foraging loads are moved upwards in the nest to be deposited. So foragers enter the nest, drop off their foraging load with a worker inside the nest. That worker will first look for a place within the brood cluster to drop off that food store. And if it can't find anywhere to do that, it'll start moving upwards to deposit that store somewhere higher up in the nest. And so what you might have noticed is that this means that the workers depositing the foraging loads and the queen are all sort of in competition for this central lower area in the colony to drop off their foraging loads or their eggs. So the reason that the brood is maintained is because food that is stored near the brood is consumed first. The bees tend to aggregate over the brood comb. Brood are really the colony's most important resource. They need to be fed. They need to be thermoregulated. So most of the bees tend to just hang out there. And it makes sense that food is consumed near where the bees tend to exist than consumed from far away. You could think of it as like if you're going out to eat, you're more likely to eat somewhere that's right down the street than to go all the way across town to eat. And sort of a fourth, I guess, hypothesized rule that I've been critically testing for the past few years is that thermal gradients affect the placement of brood within the colony. So the question that I was really asking is comb arrangement responsive to thermal gradients within the colony? So bees tend to store really more things in this, but generally 5 things within their colony. Honey, nectar, and pollen, which I classify as non-brood. They can also be classified maybe as food, as well as capped brood and uncapped brood. So old and young brood, which I classify both as food, or sorry, as brood. And for the purposes of this, we can also think of them as temperature-resistant stores. So honey, nectar, and pollen can be stored really almost any temperature and be okay. And temperature-sensitive stores. Brood can only be, brood need to be maintained at a specific temperature within a few degrees in order to develop properly. So you can imagine that brood might be very sensitive to thermal gradients within the colony. So I first tested this in 2019, last summer, or I guess 2 summers ago now. And I had 2 general groups of observation hives. So these observation hives, which are represented here, are sort of small 2-dimensional slices of what a full-size honeybee colony would be. And I had 2 separate groups. One of the groups had a heating pad on the bottom near where the brood cluster tends to be, and an unheated area on the top where the honey and nectar tend to be. And the other group was reversed with a heated section on the top and unheated near the bottom. And what I did was once a week for 11 weeks, I counted how much brood and how much non-brood was in each of these experimental areas, once per week. And what I found was that comb arrangement is indeed responsive to thermal gradients. So here we have brood cells over time in heated yellow and unheated blue areas. And you can see that after a delay, there's significantly more brood in unheated areas compared to heated areas. And for non-brood, we see the opposite effect. So non-brood over time, there's significantly more non-brood in heated areas than unheated areas. And unfortunately, I don't have a fancy publication like Angela did to show you guys, but this is in review and has been submitted. So comb arrangement is responsive to thermal gradients, and it seems to me that it seems likely that brood is driving this, that brood is being moved out of the heated sections, or brood breeding is decreased in heated sections over time. And sort of like a mechanism that I think might be driving this. So this is sort of a simplified representation of a honeybee colony with brood cells represented here in the brown hexes and non-brood cells represented here in the blue hexes. So generally, as cells are vacated, so here maybe brood emerges or maybe some nectar is consumed, they tend to be replaced by contents that were the same as what left them. Occasionally this changes, so occasionally maybe some brood gets placed where non-brood once was, but the overall structure and organization of the colony remains. But if we add a heat source, so in this case represented by the sun, and in this case a directional heat source, so it's hitting mostly the right side of the colony, we might see something different. As brood cells are vacated, they might become filled with non-brood cells as those areas become unsafe for brood to develop in. And brood breeding may shift to a different part of the hive. So in this case, we see the overall organization of the comb changing. So comb arrangement is responsive, and after a delay, less brood is present in heated sections than unheated sections. So what I was interested in looking at this year is comb arrangement resilient? And remember, resilience is the capacity of a biological system to return to its original state after perturbation. So for bees, what that means is, can comb phenotype return to its original state after being subjected to heat stress? So here I used a similar experimental design as my previous study. Again, we have our 2-frame observation hive, except in this time, instead of placing a heating pad on the top and the bottom, I had both of my experimental areas on the bottom. So for 4 weeks, I heated one area and left one area unheated. And I counted brood and non-brood twice a week this time. And after those 4 weeks, I switched the areas. So the previously heated area became unheated, and the previously unheated area became heated. With the overall hypothesis being that if comb arrangement is resilient, then the comb pattern of stress areas will recover after the stressor is removed. So this figure here is a little different than the figure before. So here we have, again, brood over time, and we have in blue the ambient areas first, which then switch to being heated, and in yellow, heated areas, which then switch to being ambient. And the switch of the temperature occurs at this, this dotted black line. And so remember, we expected that if comb arrangement was resilient, we would see that heated areas would have significantly less brood at first. This heated area would have significantly less brood and then significantly more brood when it came to ambient. That's actually not what we see. What I saw here was that the ambient area did at first have significantly more brood than the heated area, but when we switched the temperatures at this black line, the area that was once ambient but now became hot still had more brood than the area that was hot and then became ambient. So it seems like I didn't see any evidence of resilience in comb organization. What I did see, which is interesting, so if you notice the dates here, this experiment started in sort of mid to late July and ended October 1st. And what I saw was that, so generally in honeybee colonies, brood rearing decreases starting sort of in mid-August. So what you would expect is that overall brood numbers would go down. What I actually saw was that in the heated areas, the newly heated areas, brood rearing went up. So what I suspect we may be seeing is that actually this heat is increasing the length of the laying period in those heated areas. And I would expect too, so I would expect, based on what I know about honeycomb, that comb arrangement would be resilient. And I think what might have happened here is sort of because of COVID, I had to start this experiment about a month and a half later than I would have liked to. So I think if I repeated this experiment again, except I started in early June instead of in mid to late July, we might see something different. So I am excited to sort of try this again in the future at an earlier date in the summer. So we do, I guess, now have a good idea of what happens to honeybee colonies in 2 dimensions. But remember, all my studies so far have been on these sort of 2-frame observation hives, which are just a horizontal slice, or sorry, a vertical slice of a honeybee colony. Real honeybee colonies, full-size honeybee colonies, look more like this. Instead of 2 frames, they're 10 frames across with 2 of these boxes stacked high. So they're 20 frames, 10 times larger than the colonies that I was looking at before. And importantly, they're 3-dimensional. You can see here that the honeybees are spread across, in this case, 4 frames. So they really have a 3-dimensional shape to them, not the sort of 2-dimensional shapes we've been looking at before. So what I want to know now is, how does heat affect comb phenotype in a full-size colony? So again, a full-size colony, represented here as a colony sort of constrained by the size of a cavity in a tree, is arranged with honey on top, represented in blue, brood below, represented in orange, with a thin strip of pollen between the two. And remember that this is a horizontal sort of cross-sectional slice of a colony. So this honey that's on the outside of the brood wouldn't just be in the front and the back, it would actually be encompassing the entire brood cluster. What I would expect if we heated the colony is that this honey storage would sort of encroach inward into the colony. Honey provides 2 benefits. First, the immediate benefit is that because brood is dying in those areas that are maybe too hot for the brood to exist in, honey can exist there without any issue. And second, a thicker layer of honey on the outside of the colony adds some insulating benefit to the colony, allowing the bees to more easily keep the inside of their colony a temperature appropriate for brood rearing. And as the honey pushes in, what I would expect is that the queen continues to lay the same amount of brood, and that pushes the brood cluster sort of up within the colony. So you notice here that the brood cluster is taller. And just to sort of drive home that these are 3-dimensional shapes and they're not 2-dimensional shapes, in the control, the brood cluster looks sort of like a football, whereas in the heated colonies, I imagine the brood cluster will look more like a bowling pin, sort of taller and narrower. So how I'm planning on testing this is next summer, instead of using my observation hives, I'm going to use full-size Langstroth colonies. I'm going to have 2 groups. A control group that's unheated, just going to be subject to whatever the temperature is ambiently. And a heated group, which I'm going to place heating pads on the inside of and heat for 8 hours a day to simulate chronic heat stress, like a heat wave. And I really want to measure 3 things. First, does heat affect the quantity of brood and non-brood within a colony? So I'm sort of taking as an assumption that the queen is going to continue to lay the same amount of brood and that the amount of non-brood is also going to be the same over time. But you could very easily imagine a situation where maybe brood rearing is decreased because the colony is stressed, and maybe there's less honey and nectar because the bees are eating more of it in order to meet the energetic costs of increased thermoregulatory activity within the colony. Next, does heat affect the phenotypic arrangement of the comb? That's the figure I showed you guys before. And I'm very confident that it does, but I'm excited to actually look at if it actually affects the phenotypic arrangement of comb in a full-size colony. And finally, is the phenotypic arrangement of comb resilient? So what I'm going to do is I'm going to heat these for 4 to 5 weeks to let the initial change in phenotype take place, and then turn the heat off and continue to measure quantity of brood and non-brood and the phenotypic arrangement of the comb, and see if it can recover after some time being unheated. So finally, importance and outcomes. As most of you guys know, honeybees are vital for food security. Something like 1/3 of the food that we eat can be attributed to honeybee pollination. But heat stress is a continuing problem for honeybees, especially as global warming continues to worsen and nothing continues to be done about it. I think that it's really important to keep this in mind and find ways that we can help honeybees deal with heat stress, since they already have so many other issues sort of plaguing honeybee colonies right now. And what I think is really cool is that this research will result in actionable and cost-effective methods for for beekeepers to sort of maintain the health of their hives. So first, it allows to identify phenotypic markers of heat stress. So if the phenotypic arrangement of comb does indeed change due to heat stress, beekeepers will be able to actually open up their colonies, look at their comb, see how it's arranged, and see if their bees, be able to interpret then if their bees are undergoing heat stress. And then could easily move their bees into the shade or take other actions to deal with those heat stressors. And it could allow honeybee keepers to actively modify the arrangement of their comb to facilitate resilience in colonies. So if beekeepers know that maybe a heat wave is coming, instead of waiting for the bees to change the arrangement of their comb themselves, which can take a few weeks for the bees to be able to do this, they can simply, it's really easy to take frames out of these colonies and move honey frames around and brood frames around to arrange them however we want to arrange them. So you can change them to phenotypes that are heat resilient, and then when the heat stressor is going away, you can change them back to the standard phenotype, which could also just be a really interesting, cost-effective way to help the colonies deal with this heat stress. And with that, I'd like to thank the Starks Lab, and yeah, I'll take any questions.
Great, thanks, Isaac. Are there questions?
I have a question.
I don't know who's asking, but go ahead.
This is Ajal, hi. So it sounds like you were planning to, with these sort of bigger, more three-dimensional hives, add in sort of a resilience component by turning off the heat at some point and seeing if the hives return to their initial setup. Do you think there might be any value to leaving the heat on some of them to sort of test the hypothesis about sort of laying period to see if they continue to lay brood at higher rates later in the season if the heat continues, sort of like the hypothesis you kind of found with the two-dimensional hives? Yeah,
so I was actually just talking to Phil about this the other day, but I decided not to include it in this talk, but that the plan was to, at the end of the year, sort of hopefully start this in June. By July, turn off the heat, and then maybe in August or early September, turn the heat back on and see if that does increase brood rearing again in some of the colonies. So yeah, I think that is something that will be really interesting to experimentally test.
Great. Yeah, I had a similar kind of question. Just all of this depends on the ambient temperature and the changes of temperature throughout the summer. And so, you may have not just, a heat effect and maybe a resilience effect, but also sort of interactions with what's happening in the environment around at the same time. And
yeah,
so I sort of I wonder if you might set up a few different I don't know how many colonies you could set up, but a few different starts over the course of the summer to sort of
oh sure.
that kind of.
So you're saying like have some that are heated the first week and then some you delay starting the heat until like week 3 and then do it for, yeah, that could be really interesting. Yeah, that's a good idea. Because it could definitely be different throughout the summer. And like I think are getting at early in the summer, maybe it's cool. So the heat stress is going to be more of a difference from the control versus later in the summer, maybe it's just hotter that year.
Yep.
Yeah, that could be a really interesting idea. Yeah.
All right. And one quick question from Gayatri, and then we ought to move on.
Yeah, I wanted to ask if the brood, non-brood rearrangement is their first reaction to change in temperature?
Sure. So bees actually have like a lot of ways that they can regulate temperature in the hive. So for like acute heat stress, so if it's just hot for like a day, what they'll do usually is they'll fan their wings, and that can cool the colony down a lot. They can actually take water and spray it throughout the hive and use evaporative cooling, so sort of like sweat. So they have a lot of ways that they can actively cool the hive before heat stress, before changing the arrangement of the comb. That to me seems more like a response to chronic heat stress when it's been going on for like several weeks. So it's more of like a last ditch effort to control the heat, I think.
And you have only studied what happens when you increase the temperature of the hive, right? Have you tried going the other way, maybe just selectively cooling down?
Sure. I have not looked at that, mostly because it's easier to stick a heating pad on the hive and heat it than it is to find a way to keep it cold for a long time. But that would also be something really interesting to study, because honeybees are active all winter long. They don't go dormant like other insects. That would be a really interesting question.
Right.
Great. We should move on. So
thank you guys.
Patrick.